Welcome to the webinar, Using Advanced Data Visualization to Power Up Clinical Trials. Today we'll be talking about how data visualization analytics can surface valuable insights in clinical trials, insights that have the potential to save time, reduce cost, and improve patient safety across multiple phases of a trial. We'll also be giving a demonstration of clinical trial data visualization in Spotfire, a modern analytics tool. You will want to see that for sure. Before we get started, we have a few simple housekeeping matters for our viewers. At the bottom of your screen, you'll find some application widgets for your use. These widgets are all resizable and movable, so feel free to move them around to optimize your desktop space. Now, the slides will advance automatically throughout the presentation. To enlarge the slides, click the Enlarge Slides button in the top right-hand corner of your presentation window. Now, we encourage you to submit questions at any time throughout the presentation using the Q&A widget. We will try to answer your questions during the webcast, but if a fuller answer is needed or if we run out of time, it will be answered later via email. So please know that we do capture all questions and we want to make sure you receive an answer. So go ahead and submit those as you think of them during the presentation. Well, now it's time to get started. My name is Laura Beth Ezel, and I am thrilled to introduce three leading industry experts who will be joining us for today's discussion. Joanna Guild serves as the Associate Director of Clinical Data Programming at Roe. Becky Baggett is Executive Director of Project Delivery at Roe, and Jamie Chang is the Medical Director at Roe. Joanna draws on more than 20 years of experience, including 6 years of clinical data analytics expertise to lead her team in developing and employing innovative tools to manage clinical databases and identify trends in the data. Prior to joining Roe, she studied and then taught mathematics and statistics. She spent 8 years teaching at the College of Idaho. Becky Baggett has more than 16 years of experience managing phase 1 to 3 clinical trials. Over this time, she gained experience in multiple therapeutic areas, including pain, oncology, ophthalmology, dermatology, gastroenterology, organ transplantation, and allergy management. Her strong understanding of clinical trial management and delivery requirements has enabled her to successfully lead teams to meet or beat deliverable timelines in various multi-center trials. Dr. Jamie Chang is a medical officer at Roe. In this role, Dr. Chang's responsibilities include pharmacovigilance and advising project teams on clinical and scientific issues to ensure safe and well-executed clinical trials. So thank you so much for taking time to speak with us today, Joanna, Becky, and Jamie. So let's dive into our discussion here today. Trial sponsors understand clinical research is very much a data-driven process, with every phase of an investigation requiring that data be collected, monitored, analyzed, and reported. They're also aware that clinical trials tend to be expensive and inherently risky. But in today's world, emerging data science technologies can bring enormous value to clinical research, mitigating some of those risks by enabling sponsors to make better informed decisions based on timely real-world evidence. So can you tell us more about how data visualization can add value to clinical trials, and why are more and more CROs and trial sponsors turning to interactive data visualization tools to help them share and understand what's taking place during clinical investigations? Joanna, we'll start here with you.
Yeah, thanks, Laura Beth. So as you mentioned, I used to be a statistics professor, and one of the fundamental takeaways I always tried to communicate to my students is the power of data visualization. Before you do any analysis of any data, it helps to understand the data, and often the best way to do that is through visualization. And I was actually surprised when I transitioned into clinical research that data visualization wasn't even more common than it is across the clinical research industry. There has been a lot of progress just in the last several years. And data visualization is something that Roe has championed for a long time. But it was a little surprising to hear that pouring through PDF listings or Excel spreadsheets is something that happened relatively recently and still happens from time to time. So now I guess you know my kind of math nerd hidden agenda here. I'm definitely pro data visualization, but seriously, clinical research is so complex, and there are so many ways that interactive data visualizations can help support clinical trials, and we'll cover just a few today. One example, so we often see study status reports, things on study startup progress, enrollment progress, things like that. We often see those in Excel format. And Excel is very convenient for data tabulation. It has a lot of strengths, but the data are static. And so as soon as a status report in Excel format gets sent out, the data are out of date. And if that report gets forwarded on or shared with a few people in an email chain, something like that, it's really easy for the data to be like maybe nearly a week out of date by the time the report gets into the hands of a particular reviewer. So to avoid this, the way that Row has tailored our Spotfire data visualization platform allows users to log in via a web browser and access data that are refreshed typically on a daily basis. I will mention there are other visualization platforms out there. Spotfire is the system that Row has chosen to tailor for our needs. So access is internet-based, easy. Row has these visuals set up so that data are refreshed regularly, and the visuals that Row has built are not only tailored to the needs of our Row and client study team members, they're also interactive, which you often don't see in an Excel report, and you certainly don't see in a PDF report. And this really close to real-time access to data really allows the entire study team to keep a finger on the pulse of the trial. And that has many significant benefits. I'll mention two of them. One is transparency. The other is the ability to support rapid response. And I'll just kind of briefly go into detail on both of those. First, transparency. So at Row, we really value transparency. We think that there are significant benefits in making it clear to the whole team where a study is timeline-wise, so that it doesn't come as a surprise to anyone, say at the monthly status check-in meeting if things are behind. Instead, it's preferable to have any small signals of delay detected as early as possible so that they can be corrected and interventions can happen as early as possible before things potentially get out of hand and become more challenging to correct. And Becky and Jamie will talk more about what steps our Row teams take to make those course corrections early. But I want to underscore the importance of transparency at Row. Site activation is one example. Site activation is a complex, multi-step process. And using the visuals that Row has built to monitor each step along the way allows study teams to quickly identify if a site has fallen behind on one of those critical steps leading to site activation. And identifying that quickly allows the study team, again, to make those course corrections early before delays affect timelines. So that transparency, and then that second benefit is around rapid response. So, you know, we recognize that questions might come to our Row clients from their board members. And at a moment's notice, our clients need to be able to turn around that information for their executives. These web-based visuals provide that capability to access up-to-date information that you need when you need it, so that our clients have that ability to answer questions being asked by their board without needing the CRO to play that middleman and potentially slow things down. So that rapid response. And then secondly, Row is a global company. We have teams working across the world in a variety of time zones. And these web-based visuals allow team members to have the access they need to that up-to-date data, no matter what time zone they're in, whether it's business hours or the middle of the night on the East Coast of the US where Row is headquartered.
Yeah, thank you for sharing those insights there. You know, it's exciting to think about how much potential data analytics must improve monitoring and reporting in clinical research. So can you talk a bit more specifically about why Row decided to make this a company priority, and how does the data visualization platform, you know, that you've customized, Spotfire, help you to bring value to your clients?
Yeah, So I will start by saying that Roe has a really strong history of data visualization, in particular in the safety data space. Our medical monitors have been really strong proponents of the value in data visualization as a means to spot data anomalies or potential safety signals. And they truly understand how much more efficient and effective data visuals are as opposed to pouring through those Excel spreadsheets or PDF listings. And so Roe actually has been using various in-house visualization tools internally for many years, including for safety visualization. So we actually have a wealth of in-house expertise on data visualization. And so Roe decided about a year ago to really prioritize not only data visualization, but also prioritize it as an external tool and to tailor web-based visuals that are available to the Roe team and to our clients. And so we leveraged that in-house data visualization expertise as we customize Spotfire to create the visuals that you'll see Becky and Jamie demonstrate. We really wanted to ensure that what we built truly adds value to the study team. And as a result, we found that these visuals have really shifted the way that Roe does business. And again, Becky and Jamie will get into that in a little bit more detail. Roe has customized a few sets of visuals, and we started with the ones that we assessed as being most impactful. You'll see Becky share our study startup visuals, which tracks site selection, site activation, as well as subject enrollment and retention. And then Jamie will share our safety, medical monitoring, pharmacovigilance visuals. Roe has also built out patient profiles, and we're currently tailoring a set of KRIs, key risk indicators, to support risk-based quality management, RBQM. And for each of those sets of visuals, Roe has tailored the main visuals in a way for our end users to easily detect signals. And again, you'll see this in the demos. There's color coding, there's other user-friendly signaling methods. And if a signal is detected, we've made sure at Roe to make sure that the corresponding underlying data is available in the system as well, so that the end user sees the potential signal and then is able to drill down and really determine the root cause of the signal. You know, maybe the signal is a site that's not enrolling at the rate we expected. Maybe the signal is a pattern or a spike in lab test results that wasn't expected. Whatever that signal is, data visualization provides a great way for study team members, again, to easily detect that signal. We've ensured that the requisite data is available for the user to drill down to get that information that they need, because most importantly, study teams need to have the information to inform what action needs to be taken as a result of the signal that they're seeing, right? The bottom line here is that data visualizations provide information to study teams in a clear, up-to-date format, but the visualizations themselves aren't what speed up timelines. The visuals don't improve data quality. That's the study team. So the study team has that ability to ensure timelines are met, to take the actions that they need to take to potentially improve data quality. And again, you'll hear more about that from Becky and Jamie about how we use these visuals at Roe to take action in a way that can make those positive impacts on study timelines, on data quality, on site performance. Before I turn it over to Becky and Jamie, I do want to mention a final benefit of interactive data visualizations, which is the ability to connect two separate issues that may be related, but not easily connectable if data review occurs in separate spreadsheets or in separate locations. And you'll see this in the system during Jamie's demo. But one example in terms of being able to make that connection is as it applies to lab results. So if a particular lab value, say AST, for a subject spikes at a given visit, the medical monitor might want to look across data in a few different dimensions to gather relevant context. And I will preface this by saying that I am not a medical monitor, but there are three dimensions that I understand as being important. The first is to look across results for that particular subject across time. to see how significant the spike is compared to baseline values for that subject. And this applies, I mentioned AST, but it applies to heart rate, to blood pressure, to many quantitative results collected during clinical trials, right? Comparing results for a particular subject against their baseline. So that's the first dimension. The second dimension is to look across other lab results or other vital signs results for that particular subject to help assess potential medical causation. So looking if AST spiked, looking at ALT or other liver enzymes. So that's kind of the second dimension for that subject, other results. And then the third dimension is to look across, again, for example, AST results for other subjects across time, right? To see if there's some study-related pattern. Is there a collective spike or collective dip in values you see for AST, for blood pressure, et cetera, after visit 4 or after a third dose? And all of that is so much easier to do visually than across spreadsheets or within listings. So again, Ro has really tailored our suite of medical monitoring visuals to allow our medical monitors to view the data in each of those dimensions and then some. And at this point, I'll stop kind of talking theory. And Laura Beth, can we turn it over to Becky and to Jamie to actually kind of share some of this with the audience?
Absolutely. And you've done a great job there setting us up. So thank you for that, Joanna. And I'm looking forward to seeing this data visualization platform in action. So our presenters are going to be using the media player widget for the demo here. So to improve your viewing experience, you can click on the button with the 4 arrows to expand the media player and follow along with the demo as we go through it here. We just want to let our audience know that so you can have the best viewing experience. So Becky, I'm going to turn it over to you to get us started with the first demo.
Thank you so much. And I get the fun job here today. I know you heard a lot from Joanna about what thought has gone into the design of Spotfire for Ro's dashboards. And really today, my hope is to give you an inside look of how we use these dashboards to do our own signal detection, our monitoring of the study progress overall, looking for little clues of places that we might need to correct course sooner rather than later. Sometimes we're just looking and we're watching, but sometimes we know we need to take action before things get worse. So as you see on your screen, I want to just orient you quickly to what you're looking at. There is a disclaimer at the top that explains the things that Joanna was referring to. These graphics are refreshed about every 24 hours. Right now, we're looking at a demo system. It pulls from our EDC system as well as our CTMS system. And it will give you in this disclaimer the data cutoff date, which for this demo study is December 1st of 2020. So that will give you a frame of reference of time, which is important while you're reading through these graphics. It's also important to note that the data pulls straight from these systems, and it's dirty data. So while it's great and it's informative, sometimes it can be data entry errors that we're seeing. And we want to make sure that we understand what the correct data is before we take true action and go down the wrong path. So just a few things to keep in mind. And as I go through this, I'm going to sort of talk out loud to give you some examples of places where we have used these dashboards to take action during the study. And that's from site selection all the way through subject discontinuation. So let me start by looking at our first of 2 executive dashboards, executive summaries. Across the top, real quick, you can see purple bars represent the site data. So we have 34 out of the 35 sites selected for the study. 24 out of those 35 have been activated. Gold bars are going to represent subject data. So we have 175 subjects out of the 350 projected screened, 139 randomized out of the 280, that's our goal. 12 have discontinued, 41 have completed, and 11 have discontinued treatment. So again, very quick, high levels executive summary. This is the second of our executive summaries, very high level view. Again, these tiles on the left are going to remind us of the metrics that we viewed on the previous screen. But as you can see, this is now showing enrollment and screening actuals and projections over time, laid on top of our site activation actuals and projections over time. The darker colors represent the actuals. The lighter colors represent our projections. So very simple. Most people have seen these types of graphs. They're used constantly in clinical trials. So this is where the signal detections begins for us. So when I look at this, I see 24 sites out of the 25 we projected were activated in the month of November. I know my data cut is December 1st, so November has been completed. So my question is why? Why do we have 24 out of the 25? Knowing that the data was cut on the 1st of December, it could be that the data entry timing didn't quite make it in for that December 1st cut. And when I look tomorrow, it'll be 25 and 25. It could also mean that one of the sites had to go for a second review with the IRB. Maybe something changed with the consent form, and now that site has been pushed into December for activation. And these are all questions I'm following up with my startup team to determine. You'll also notice here with this dark color, the screening, dark blue actual is trending a little bit higher than the projection we have in the light blue, while randomization in the dark orange is trending a little bit lower than the projection in the lighter orange randomization. Not huge swings, and we know that nothing's linear in clinical trials, but I am going to keep an eye on it because it could mean that our screen failure percentage is increasing, and I want to know sooner rather than later. So moving through, we're going to get to now the real details of the study and the management, so we can see through the progression from selection until subject discontinuation, monitoring these activities and where we look to take action. So just to orient you again, the tiles on the left are going to give you a summary of site status. So we look at identified, CDAs executed, qualification visits either completed or waived, and then how many sites we have selected. We use stoplight colors here in the table, as you can see, to very easily identify progress with the all green, work in progress in yellow, and red being a hard stop. And you'll notice that red for us here, we're using it with the sites that weren't selected. So if I want to get a more streamlined view, I can very quickly, using my filters over here, remove the ones that were not selected. And now I have a cleaner view of those sites that are actually in progress or have been selected. In the yellow progress bars, these will help me figure out where I'm seeing things stall. So as you can see, these 5 sites have kind of stalled with CDAs. Now, for the ones that were identified in November, probably not a big deal. When I'm looking at the one in September, I'm really wanting to know why. Why is it now December 1st and I have not had a CDA executed with the site that was identified back in September? This is a question I'm going to go back to my startup team with to find out what's going on and push this through. You know, Joanna talked about transparency with these graphs, and truly what you see here is what I see, is what my client sees. And it will show us where we might have gaps in our processes. So you'll notice site 80 here, 2 items are yellow while they've been selected. This will make me go back to the CTA who is involved with site 80 and make sure that they are keeping their data entry in real time up to date. So again, some things are site issues, some things are process issues, but you can really quickly see when you're looking at these graphics. And then just one last example, if you want a quick list of your selected sites, you can easily do that with the filters as well. So now that we've had our sites selected, we're going to check on their activation status. This should be a very familiar graph with the site activation over time. Actuals are in dark colors, projections are in the lighter colors over time across the bottom here. These bars over here, the dark purple, represent the steps that sites need to go through in order to become activated for a study. So we can see in each bucket how many fall from the sites that we have selected for each bucket. And for me, when I'm looking at this graph, very quickly I can see I've got 24 activated, but I have 26 that have made it through all the steps. So if I click on this SIV bar, the table down here will filter and show me immediately site 23 and site 34, they're ready and they need to be activated today. So that's the message to my team in the meeting. 23 and 34, get activated today. So really quickly, you can prioritize your sites by just clicking on these filters here, filtering in the table. It's super useful when you're trying to prioritize, which is a constant juggle. Those of you who have done it from month to month for that activation, it really makes it easy versus filtering and sorting in an Excel spreadsheet. So now that we're through activation, we're going to go back and we're going to look at enrollment. And this will be a familiar graph that you saw in the executive summary. But this has a little bit more functionality so that you can dig in a bit more on the enrollment. So as you see here, again, site activations, and then screening and enrollment laid over top over time. So what you can also do in this dashboard, we've set it up so we can review previous projections. So let's say I want to take a look at when we started back in August and see what we had originally projected. You'll notice that over here, we were projecting that enrollment would end in March. So through an amendment or maybe an advertising campaign, things started going really well, as you can see the progress, and we ended up reprojecting. So here we're sitting December 1st, and now you'll notice, now we're projecting February is when we will end enrollment. So it's really nice to be able to show that level of progress through time. You'll also see that there are the subject information, high-level subject information listed in this table before. Down below, you can actually click on a site and it will filter the table for that site's information. And so you can also select or multi-select. Let's say you have 3 sites in a network that you're wanting to take a look at, make sure that they are performing because they promise big numbers. And so now we're going to make sure that they're following through on that promise. Well, let's see, site 6 and 13 look pretty good. But we get down, we see 25, they've got 1 screened and 1 randomized. So that's not the same as a comparison with their colleagues. So I'm going to make a note, site 25 is going to go on my list here. And as I mentioned before, you know, the data, it's a signal, it doesn't necessarily mean a problem. And I'll give you an example in a minute of what I mean. So now I'm going to move through to subject enrollment. So looking on the left, again, the blue tiles giving you a reminder of what we saw on the executive summary. And we move through to randomize versus screen failed by site. This graphic here, it should be a somewhat predictable pattern in terms of number of subjects randomized, which is represented in blue, and then the number of subjects screen failed, which is represented in orange. We know what our screen failure rate is for the study, so there should be a predictable general pattern that you're seeing here with these ratios. This helps you quickly identify where that pattern isn't quite lining up. So for example, site 30 here, they have screen failed 3 subjects and randomized 2, which is not expected. So I will make a note of site 30. I would like to dig in a little bit more to find out what's going on there. Other sites, patterns that we would want to investigate. If you look at 64 down here, well, the long blue bar looks great. They've enrolled 15. However, they haven't screen failed anyone, which is curious. I'm not saying it's a problem, but I think you're going to want to ask your CRA to take a closer look at their screening and enrolling practices, make sure they understand the protocol, the inclusion-exclusion criteria, so that no subjects are enrolled that are not appropriate for the study. Could be that they've got it down great. This is a very experienced site, but this is just a little piece of data to help you figure these things out and prevent problems from happening. If we move to the right, this next graph with the purple bars is going to show you days since last screened or last randomized. And right now it's filtered to randomized, but very quickly you can identify problem areas. So this site here, 30, we talked about them before. When I click on this bar, it will isolate them in both graphs. And then I can hover over, and it's going to tell me that it's been 29 days since they've last randomized someone, which is almost a month. But it's been 2 days since they've last screened. So this tells me that they might be struggling a little bit with identifying the right subjects, but they're trying because they've been screening as recently as 2 days ago. So I'm going to still keep them on my list. This is now additional information that I jot down to make sure that I follow up about. Now we're going to go and look at the site that we identified before, 25, in the previous graph. When I click on 25, it hasn't been a whole lot of time since they've screened and randomized. It's only been 2 days since they've randomized someone, so that's actually not bad. I can come over here and see that they've actually randomized one person. So now I'm curious, when did they get activated? So when I go back to the site activation tab, I can very quickly in my table look and see, okay, they were activated on November 27th, very recently. This site is fine. 25 comes off my list. I don't need to go any further because I think what they're doing looks good to me. So you may have noticed that over here, these tables here will filter the screen failure data based on which site is selected. So again, looking at site 30, I quickly have the IDs of those subjects that did screen fail, and I can see if it was that they withdrew consent or if it was because of inclusion and exclusion criteria. So now, if I want to dig in a little bit more about the inclusion and exclusion criteria, here I'm going to figure out why subjects are not making it into the trial. And looking at this graph, you can see that exclusion 12 and inclusion 3 are the two top reasons that subjects are not making it into the study. 7 subjects for each of these have not made it into the study. And so maybe this is expected, but let's say inclusion 3, it really wasn't expected. You can see very quickly too that there's this very long yellow bar here. What does that mean? When you click on it, it'll show you that 5 out of the total 7 have actually been at site number 42. So this tells me this is not an issue with inclusion 3. This is probably an issue with site 42's understanding of inclusion 3 or being able to identify subjects that can meet this criteria. So now when I call site 42, I have very specific information to talk to them about. How's it going? Talk to me about inclusion 3. What struggles have you seen here? And I know that it's not a study design issue with inclusion 3. We look at our site 30 that we talked about before. Now when I call them, I can say, hey, I noticed that you have had, your subject screen fail in exclusion 27, as well as inclusion 5. Let's talk about these. Let's see if we could get you a better pre-screener. What challenges have you seen? It's a much more robust conversation, whereas you already have the information at hand and you don't need to ask the site to go digging through their files or recapture, recount all of the information that you now have already gained through these graphics. And again, you can see that there is filtering in the table over here based on which color you're clicking to the right. So moving through enrollment, now we're looking at subject disposition. So again, we have our tiles here that are reminding us of what we saw in our executive summary. And what you'll notice is we also are using the stoplight colors that we discussed earlier with site selection in our representation of randomized to discontinued ratio. So the ratio, the perceived negative ratio is red, and then gradually moving towards bright green based on the calculated ratio. So here we are again with our friends at site 5. So we know that they have had a long time based on this site enrollment graph. They were the longest one. It's been almost 2 months for this site that they haven't screened or enrolled anyone. So now we really are curious because their one and only subject has discontinued. And oh, by the way, they withdrew consent. So this gives us a lot of information to go on. And maybe we knew that this site was going to be inactive. Obviously through the last 2 years, we saw a lot of sites closed down for COVID. So the CRA told us, we knew it would be a delay, but was it a 50-day delay? Should it have only been 30 days? These are the questions that we're going to go back to the CRA and then follow up with the site to see what's going on here. Looking at, let's say, site 13, 3 discontinued, that's the highest number at a site for number of discontinued subjects. So you can see very quickly the IDs that are associated with this ratio. And what's more interesting is you can see when they discontinued. So 2 out of 3 were in the first week of the study, which is a little concerning. So we're certainly going to want to follow up and find out what's going on there. You can also see this represented down here with these darker dots. These are the subjects that are listed in the table above at site 13, showing where they have discontinued in relation to the rest of the study subjects. The other really useful piece of this graph right here, honestly, I feel is one of the most important, is that if you, for example, my study, its primary endpoint lands on day 30. If I look at this graph, more than half of the subjects who have discontinued from the study have discontinued before my primary endpoint. Now, this might point to a design issue that I'm definitely going to want to discuss with the sponsor, because we would want to correct course and nip this in the bud before it became a larger problem for the study. So these are just some very quick, high-level examples of how our project management and ClinOps team use these dashboards, and there's obviously other dashboards that we use to monitor the study, share this information with the sponsor, and have just much more robust discussions with the team, with the sponsor, with the sites, and everyone involved. It's a much more collaborative discussion, and we feel it's a much easier way to manage the trials and detect those signals and make changes, quite honestly, that probably should happen as soon as possible. So with that, I'm going to go ahead and turn it back over to you, Laura Beth. I hope that everyone was able to enjoy the presentation, but definitely open for questions at the end.
Absolutely. Thank you, Becky. That was very thorough and easy to follow, and really appreciate you putting that together for our audience. So it's exciting to see just how much easier data visualization can make, some of these challenging aspects of site management. It was neat to just see you dig into it and find answers. So now let's give the floor to Jamie, and he's going to demonstrate how Spotfire can be used to perform medical data reviews. So Jamie, we're going to hand it over to you and let you walk us through our second demo of this presentation.
Great. Thank you, Laura Beth. Thank you, Becky. I think, as Joanna has described, Spotfire is an extremely versatile platform to visualize data and to act on it in a very proactive way. And that certainly applies to safety. I think we've mentioned several times that we really believe this is a great way to enhance signal detection, whether that's sort of in terms of clinical operations or in terms of safety. The other part that we really want to emphasize is it really allows all the functional areas who are using the tool to participate in the process of data quality review. We're constantly looking for an indication that certainly in safety, for safety signals, but also is there some indication that the data is being entered in such a way that it might be erroneous or might contaminate the overall trial data. So that's a real advantage. And what I hope to do in the next couple of minutes, and this is the latter part of our webinar, is show you, and I'm going to share my screen here, show you through the safety visuals how some of these principles come to life. So we will start with, again, as Becky pointed out, some of the key things. When the demo, this is a demo study, so this is demo data, and it sort of tells you when it's most current. So when you're looking at safety data, you know when it was last updated, which is obviously very key when you're looking over this data. The first module in Spotfire safety is called the Adverse Event Summary, and it sort of it says what it is, but very quickly you can see here Adverse Event Summary, you get a very quick qualitative sense of the overall adverse events that have occurred in your study to date. These are preferred terms, but you can ask, based on the population in your study, based on your investigational product, qualitatively, are these events, do these events seem like events that you would sort of expect in the study? You also can look right next door and look at a quantitative breakdown of these different AEs and say, okay, you can look at a subject percentage. So how many subjects have experienced at least one AE, then the AE percentage, how many of that particular preferred term, what percentage of all AEs does that compose? So it's a very quick way to sort of say, qualitative and quantitatively, what's the AE profile look like in our clinical trial, and does that make sense based on what we know? And very quickly, there's also other ways that Joanna pointed out. Very quickly, I want to know other information. What's the severity look like? And with a very quick change, we can filter by severity, and we have a nice color coding here. And very quickly, you can determine, okay, this is the majority are mild events, but there are moderate, severe, and life-threatening events. So obviously of great interest. And back to the sort of the efficient use of this platform, I can click on this. So this is a grade 3 event. I can click on this event right here. And it brings up, in details on demand, it brings up the details. So the site number, I get an idea of the demographics, their age, gender. And I can also see, the terms, and importantly, what's the relationship to the drug product, and what the outcome. So very quickly, without going to another, going into EBC or something, I can drill down and understand the details of that event. And if the term seems odd or something seems odd in the details, I can reach out to the site and we can have a discussion right when I'm looking at that as well. So again, that proactive approach to making sure that these AEs are being recorded accurately, but also that we're making sure that we're understanding any signals that are arising in the study in real time. Becky pointed out that this is not, the data is, not fully clean, but certainly, and we're going to verify these things. But this is in safety, we're looking for any indication that there's a signal or something, and we have a very quick way of doing that with the adverse event summary. Another question that comes up sometimes is, well, what's the adverse event rate? What does it look like, and is it differ between sites? Are there sites there are a lot of events or very few events? That could be a safety issue, right? If there are a lot of AEs than we're expecting, or there are very few, that could be a reporting issue. But we want to know those things for safety reasons, but also for data quality reasons. So if we go to our adverse event by site module, and I'm going to orient you here a little bit. So on the y-axis is the adverse events per total subject weeks. So, one subject in for a week is a subject week. As we go to the right on the x-axis, that's more subject weeks. So we have more subjects who are in longer in the study, and we're accumulating more AE data, certainly as we go over here. Very quickly, the program sort of draws an average line of the AE rate. We can sort of see which sites may be above or below that line. And again, that could be lots of things. That could be data entry. That could be over reporting. That could be a safety signal as well. But the nice part of this is I can hover over this value. I can see what site it is, and I can go back to the adverse event. This was site, let's see, site 5. You might not be able to see that, but I can go very quickly back to my summary and say, okay, let's look at site 5. They had a fairly high AE rate, and get a qualitative sense of what sort of AEs they're reporting. And again, so to see if that sort of seems reasonable based on the number of subjects they have and the other AEs we've seen in the study. So again, a very, very quick way to get a quantitative and quantitative sense of the AEs in your study to date, and ways to zoom in fairly quickly and determine sort of if this is something that needs to be acted upon on a more urgent basis. Another question that comes up often with adverse events is, well, when are they occurring? And how long are they lasting? It's important for subjects and investigators to know this information, especially for those who haven't yet enrolled in the study. And right now, this looks extremely busy. But let's say, for example, we know that our drug may have a high rate of nausea. So I'm going to select the gastrointestinal disorders here on the filters over here. And I'm also going to select nausea. So essentially what I see here now are the preferred term of nausea, and we're looking here. And then I can tell you in this study that the dosing is done every 7 days. So we're seeing an adverse event of nausea every 7 days. which is associated with dosing. That seems to, this is the start and the finish. The start and finish arrows are sort of close together, but very short duration. So we have a sense that the nausea is of short duration, seems to occur on dosing days for some subjects, but seems to be very short duration. And we also know based on color what the severity is. Most events seem mild. Now, in full disclosure, this is demo data, and you're seeing dosing data here, which we wouldn't see in a blinded study. But again, you see an increased dose with an increased severity of the AE rate. So a really nice way, this timeline to see of sort of when these events are occurring, how long they're lasting, and sort of what the severity is. Very helpful information as you're starting to learn about the safety profile of your investigational product. We talked a lot about quality, data quality. That's in addition to the data visualization. Roe was founded as a data management company, so we are absolutely looking for data quality issues. In addition, that includes a medical data reviewer. So here we're looking at the histogram module, and again, this gives us an idea of what's the distribution of the data. And this is based on the premise that you have enough observations in a population that you should see a certain distribution of the data. A lot of times we talk about normal distributions as well. So here we're looking at heart rate, ECG heart rate, and again, sort of centered, in the 60s or so, and then we have HTAL, and that seems like a very reasonable distribution. Let's look at something like respiratory rate, which I think we all know is a vital sign that may not always be recorded with the most fidelity, just because you have to wait 30 seconds or you have to wait a minute. And sometimes people are very busy, and that's a lot for them to wait. So as we look across data, we see here over in the far left, 840 observations for 147 subjects. So that's sort of distributed in these different buckets, if you will, and it seems very reasonable. Sort of that variability based on those observations. So let's go let's go site by site though. And Becky kind of did this with the sort of looking at sort of the operational thing, but let's site by site. And here we have 12 observations with one subject, and it's very variable. So it seems within sort of statistical reasonable. And then we go to site 6, and they have 26 observations for 3, again distributed over these different rates. And then we go to site 7. And this is interesting, right? So now we see 27 observations in 8 subjects, only in 3 buckets. That's certainly possible, but we start to wonder maybe that variable is not being recorded as the protocol specified. And this is, again, in terms of, we talked about the actions that we take. We reach out to our CRA or the site, and we say, okay, let's see how the site is recording this to make sure we remedy any issues with data capture or data entry earlier before that causes any issues down the road. This is also going to prompt us to look at the other variables. So maybe I'll look at weight. Maybe I'll look at their diastolic pressure and just see if the measurements make sense in terms of the number of observations and how they're distributed. So a key part of the visualization, again, is looking at how the data is entered, and if potentially it might represent an issue with how the data is being entered and something that we need to intervene on in a timely fashion. The next module that I want to talk about, Joanna had alluded to this. This is essentially a way to put any sort of a continuous variable sort of in context. And let me explain what that means a little bit. We're looking at an ALT here. These are the values on the y-axis, the visits on the x-axis. These are individual subjects that are, with these points and these lines. They're all the subjects in the studies, and you sort of have these, you can see these dashed lines, the standard deviations, upper and lower limits, but you clearly have values, especially this one up here, that's extremely elevated. So, we quickly can put this in context. Is this a data entry error, or is this something else that could be a safety signal? And as Joanna alluded to, I click on that value, and it instantly, these details on demand, again, this information you don't have to go back and look at the lab portal, it's right here. You can see that in addition to the ALT, the bilirubin's elevated, and so is the AST. So this looks like to be something hepatic injury. So this very quickly allows us to zoom in and say this looks like a safety signal versus somebody potentially entering at a heart rate of 200 and not seeing blood pressure change or anything along those lines. So again, very quickly we can put these values in context and know if it's something that that could just be a data entry error or could be a safety signal that we need to zoom in on. Joanna also mentioned sort of the dimension, looking at a different dimension. So this is AST. We can look at all values across the study, so all AST values. So I'll just do that right now. And so we can look at this, and this represents sort of the values across the study, different visits, and the number of subjects as well. And we can see if there's any change overall in that value. In this case, I don't really think there is, but we can see if there's any trend overall. And maybe if we saw a lot of those subjects in that individual subjects, we would see a change here as well. So it's sort of a redundant way in some ways to sort of see if we can pick up if there's a safety signal that we see in the individual safety over time. Maybe that's going to manifest over across the group as well. So that's very handy to pick this up very quickly. If you look at the far right, you see the value looks a bit higher, but then you realize it's only 13 subjects for unscheduled visits as well. So again, all very accessible information. You know the number of subjects you're looking at, the values, all accessible to you right here, where you can sort of make initial conclusions and sort of pursue what you need to based on that information that you have. The last module I want to share is called Hepatic Safety Explorer, and because of the importance of hepatic signals in drug development, this is sort of a way, this will pick up Hy's Law's cases, so when there's liver injury or Temple's corollary. I would say that between the other modules, especially the individual safety over time, we should be able to pick these up. But this is a redundant way of saying there's a subject who may meet the criteria. They've had elevated AST or ALT and bilirubin and OCPHOS. And again, we can find the subject very quickly amongst the other ones and reach out to the site and investigator about this as soon as we see it. So that is a sampling of some of the modules in Spotfire safety. Again, I firmly believe this enhances signal detection, and it really allows for a very efficient kind of quantitative and qualitative decision making. Mostly by having all the information within one source and being able to drill down, as Joanna pointed down, and make the necessary conclusions and think about your next steps and how you're going to address the issue at hand. And I also just the last comment I want to make is Joanna mentioned there are patient profile in Spotfire as well, and sometimes those are used for individual subject data as well. So an extremely versatile platform, if we haven't said that enough, that can really help facilitate efficient and high quality clinical trials. And with that, I will turn it back over to Laura Beth.
Excellent job there, Jamie. Thank you all again for your presentations here. And we've been talking about and seeing firsthand how data visualization can help CROs and trial sponsors better manage sites to increase efficiencies and keep the trials running smoothly. But we've also seen another potentially pivotal application of this technology. Monitoring patient safety and watching for adverse effects has long been important in clinical trials, but reviewing thousands of data points and safety markers manually or with traditional reporting methods is inefficient. There's always a risk that clinically relevant signals might get lost in the large volumes of data involved. Interactive data visualization tools can make it much quicker and easier to identify these anomalies. This is something that's likely to be of great benefit in the future for pharmaceutical companies and the patient community alike. So thank you all. We are nearing the end of our allotted time. Before we go, we have been taking in some questions from our audience. You can continue to do that using the Q&A widget, and we'll try to get through as many as we can before our time limit here. So I'm going to jump right in on the first one here. And it's the question is, are these data visualizations an extra add-on service with Row, or are they included as standard? Who would like to take that question on for us today?
I could take that, Laura Beth. Thank you. Great question. And this is basically how Row does business. These come with all of our project management, ClinOps teams. I think if they tried to not have them come with, there'd be a small riot. We love using these tools. They obviously make our jobs much easier, much more efficient. And so this is just kind of how Road does business. So yes, they're all part of the package. This is not an add-on service, add-on feature. It's part of how we manage trials.
Excellent. Thank you for clarifying that for our audience. Okay, next question here. You mentioned data are typically refreshed on a daily basis. Do you recommend reviewing the visuals on a daily basis? Another good question here. Who'd like to take that one?
I can take that one as well. Thanks. Yeah, that is a great question. Honestly, it really depends on the study. If you have a study that is intended or it is planned to be slow rolling, that it's, going to be maybe 0.2 subjects per site per month. Daily review probably doesn't make a whole lot of sense. Maybe even some cases, weekly review may not make a whole lot of sense. So we really have to take careful consideration and discussion with your team and your sponsor to understand what the expectations overall are for the study, and then agree on the proper time points to review this. And obviously the review together is the most beneficial. So making sure everyone's on the same schedule, that they're all looking at the same data and have the same understanding of that data.
Yeah, and if I could just add to that, thanks, Becky. I think another especially from a safety perspective, what's the data of interest? Especially as we noted, if you're updating EDC data nightly or very frequently, sometimes it's just as a practical matter, your vendor data might not be coming in as frequently. And actually, we know that the lab transfers can vary every month, every couple of weeks, but it really depends what you want to look at. If you need to have the whole context, you need the vital signs and the labs together, then you're going to schedule those reviews in terms of efficiency. If you're looking at a therapy that potentially lowers blood pressure, then you can look at the data fairly often. Also, stage of development, right? I mean, if it's a first-in-human study, potentially you're going to be looking at vital sign data fairly quickly. I think the important message is you're going to have data there, at least the EDC data, on a regular basis that you can view. And if you need more, just being proactive and thinking about how to get that data into Spotfire in the timely fashion, so that you and the internal and the external teams can review that simultaneously.
Great point, Jamie. Thanks.
Thank you.
Yeah, thank you both for taking on that question there. Okay, another question here. Do these visuals require SDTM or other data standards? I'd like to tackle that one.
That's a good question. I can take that one. The visuals can certainly be built with SDTM or ADAM data, but they don't require it. Something that we found at Roe is that those standard data, SDTM, ADAM data, often aren't available as early in the study as needed in terms of when the study teams would like to begin using the visuals, especially the ones that Becky shared with you around study startup. So we've engineered things behind the scenes to allow for flexibility. There's a general format that we kind of wrangle the data into, but we do typically work from raw EDC data. And as Jamie mentioned, that allows for those daily data refreshes for data coming in from EDC, whereas SDTM or ADAM data sets are typically updated much less frequently.
All right. Okay, and another question here. What options are there to export visuals or data from the system?
I can take that one too. Spotfire offers several export options by default. All of the components you all saw can be exported as images or as PDFs. And the tables, for example, some of the site selection tables that Becky shared, all of the tables can be exported as CSV files compatible with Excel as well.
Excellent. Thank you guys for tackling a few of those before the end of our time here. Before we sign off, I just want to thank you again, Joanna, Becky, and Jamie, for taking the time to share your expertise and insights with us today. We are so grateful to have had you with us today. For our viewers, if you asked a question during the Q&A that we didn't get to, the Roe team will deliver an answer to you via email. Today's webinar will also be available on demand if you want to review anything that we've talked about today. In the resource section, you can also find a copy of the slides from today's presentation, including some additional slides, that supplement today's demos. So thanks again for watching and have a wonderful day.